Golgi apparatus. Camillo Golgi, 1898, first observed densely stained reticular structures near the nucleus. They were later named Golgi bodies after him. They consist of many flat disc shaped sag or cisternae of zero point 0.5 nanometer to 1 nanometer diameter. Figure 8.6. These are stacked parallel to each other. Various numbers of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cysts or the forming face and concave trans or the cysts or the maturing phase. The cysts and the trans phase of the organelles are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the ear fuse with the safe space of the Golgi apparatus and move toward the maturing phase. This examples why the Golgi apparatus remain in close associations with the endoplasmic reticulum. A number of protein synthesis by ribosomes on the endoplasmic reticulum are modified in the system of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formations of glycoproteins and glycolipid lysosome. These are membrane-bound vesicle structures formed by the process of packaging in the Golgi apparatus. These isolate lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolase, lipase, protease, carbohydrates, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrate, proteins, lipids, and nucleic acid. Vacuoles. The vacuoles is the membrane-bound space found in the cytoplasm. It consists of water, sap, excretory products, and other material not useful for the cell. The vacuole is bounded by a single membrane called a tonoplast. In plant cells, the vacuoles can occupy up to 90% of the vacuum of the cell. In the plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion in many cells. As in protists, food vacuoles are formed by engulfing the food particles. Mitochondria. Mitochondria, single mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activities of the cells. In terms of shape and size, also considerable degree of variability is observed. Typically, it is sac shaped or cylindrical, having a diameter of 0.2 to 1 micrometer, average zero point 0.5 micrometer, and length 1 to 4.1 micrometer. Each mitochondria is double membrane bound structures with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments in outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer matrix forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfolding called the sister cristae. Single crista toward the matrix. Figure 8.7. The crista increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouse of the cell. The matrix also possesses single circular DNA molecules, a few RNA molecules, ribosomes, 70S, and the component required for the synthesis of protein. The mitochondria divides by fusion. Plastids. Plastids are found in all plant cells and in iglonide These are easily observed under microscope as they are large. They bear some specific pigment, thus imparting specific colors to the plants based on the types of pigments. Plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat soluble carotenoid pigment like carotene, xanthophyll, and other are present. This gives the parts of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastid of varied shape and size with stored nutrients. Amyloplasts store carbohydrate, starch, example potato. Elioplasts store oil and fats, whereas the eleuroplasts store proteins. Majority of the chloroplasts of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-shaped organelles having variable length 5 to 10 micrometer and width 2 to 4 nanometer. Their number varies from 1 per cells of the Calamadomus, a green algae, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound of the two. The inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Figure 8.8. Thylakoids are arranged in the stack like the piles of coins called grana, singular granum, or the intragranular thylakoids. In addition, there are flat membranous tubules called the stromal lamella connecting the thylakoids of the different grana. The membrane of the thylakoids includes a space called a lumen. The stroma of chloroplasts contain enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller 70s than the cytoplasm ribosome 80s. Ribosomes. Ribosomes are the granular structures first observed under the electron microscopes as dense particles by George Palade, 1953. They are composed of ribonucleic acid, RNA, and protein and are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, large and small subunit. Figure 8.9. The two subunits of 80s ribosomes are 60s and 40s, while the uh, of 70s ribosomes are 50s and 30s. Hence, yes, Seuberg unit stand for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70s and 80s ribosomes are composed of two subunits, cytoskeleton, and elaborate network of filamentous protein near structures consisting a microtubule, microfilaments, and intermediate filaments present in the cytoplasm in collectively referred to as the cytoskeleton. The cytoskeleton in all cells are involved in many functions such as a mechanical supports, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia, single cilium, and flagella, single flagellum, are hair-like outgrowth of the cell membrane. Cilia are smaller structures which work like oars, causing the movements of either the cell or the surrounding fluid. Flagella are comparatively longer and are responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of a cilium of the flagellum show that they are covered with plasma membrane. Their core, called the axonema, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets or radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonema microtubules is referred to as the 9 plus 2 array. Figure 8.10. The central tubules are connected by Bridge end is also enclosed by a central seat which is connecting to one of the tubes of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnecting by linker. Both the cilium and flagellum emerge from centrole-like structures called the basal bodies. Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called a centriole. They are surrounded by a amorphous pericentriole materials. Both the centrioles and centrosomes lies perpendicular to each other in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibril of tubule protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central parts of the proximal region of the centriole is also proteinous and are called the hub, which is connecting with tubules of the peripheral triplets by radial spoke. Made of protein, the centriole form the basal body of cilia or flagella and spindle fibers that gives rise to spindle apparatus during cell development in animal cells. Nucleus. Nucleus as a cell organelle was first discovered by Robert Brown as earlier as 1831. Later, the material of the nucleus stained by the basis dyes was given the name chromatin by Fleming. The interface nucleus of a cell when it is not dividing has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one of one or more spherical bodies called a nucleoli, single nucleolus, figure 8.11. Electron microscope has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passage through which movement of RNA and the protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the numbers of the nuclei are also frequently observed. Can you recollect name of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. Example: erythrocytes of many mammals and slew tube of cells of vascular plants. Would you consider these cells are living? The nuclear matrix or the nucleoplasm contain nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm. As it is not a membrane-bound structure, it is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interface nucleus has a loose and indirect network of nucleoprotein fiber called chromatin, but during different stages of cell division, cell shows structured chromosome in place of the nucleus. Chromatin contain DNA and some basis protein called histone, some non-histone protein and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 23 pairs chromosomes. You will study the details of DNA packaging in the form of chromosome in class 12. Early chromosome, visible only in diving cells, essentially has a primary constriction or the centromere on the side of which disc-shaped structures called a kinetochore are present. Figure 8.12. Centromere holds two chromatin of a chromosome based on the positions of the centromere. The chromosomes can be classified into four types. Figure 8.13. The Metacentric chromosomes has middle centromere forming two equal arms of the chromosomes. The submetacentric chromosomes has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosomes, the centrosome is situated close to its ends, forming one extremely short and one very long arm, whereas the telocentric chromosomes has a terminal centromere. Sometimes a few chromosomes have non-standing secondary constructs at a constant location. This gives the appearance of small fragments called satellites. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plants and animal cells. Summary: all organisms are made up of cells or aggregates of cells. Cells vary in their shape and size and activities functions based on the presence or absence of membrane bound nucleus and other organelles. Cells are hence organized can be named as eukaryotic or prokaryotic. A typical eukaryotic cells consist of a cell membrane, nucleus and cytoplasm. Plant cells has a cell wall outside the cell membrane. The plasma membrane is selective permeables and facilitate transports of several molecules. The endomembranous systems includes ER, Golgi complex, lysosomes and vacuoles. All the cell organelles perform different but specific function. Centrosome and centrioles form the basal body of cilia and flagella that facilitate locomotions. In animal cells, centrioles also form spindle apparatus during cell division. Nucleus contains nucleoli and chromatin network. It not only central control the activities of organelles but also play a major role in heredity. Endoplasmic reticulum contains tubular or cristae. They are of two types, rough and smooth. ER help in the transport of substance, synthesis of proteins, lipoproteins and glycogen. The Golgi body is membranous organelle composed of flattened sac. The secretion of cells are packed in them are transported from the cell. Lysosomes are single membrane structures containing enzymes for digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. This occurs freely in the cytoplasms or are associated with ER. Mitochondria help in the oxidations, phosphorylations, and generations of adenosine triphosphate. They are bound by a double membranous. The outer membrane is smooth and inner membrane one fold into several cristae. Plastids are pigment containing organelles found in plant cells only. In plant cells, chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana is the plastid in the sites of light reactions and the stroma of dark reaction. The green colored plastids are chloroplast while the containing chlorophyll, whereas the other colored plastids are chromoplast, which may contain pigments like carotene and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pore. The inner membrane encloses the nucleoplasms and the chromatin materials, thus cell the structural and the functional unit of life.